Hello and welcome to Beyond Biotech podcast number 26 as we hurtle toward the holiday season. And as there are 52 weeks in a year, that means we've done half a year of podcasts. I'm Jim Cornell from Labiotech, and it might be the most wonderful time of the year, but it seems like it's also the busiest. This week I'm doing two kinds of interviews for reporter positions here at the publication, and I'm also doing interviews for the podcast. On top of that, we had a special newsletter on startups to do. There are people away on holiday, the news is crazy one day and then there's not much the next. And then there's that awkward week between Christmas and New Year to navigate when everyone's away and there's no news at all. We're also more than halfway through the Advent calendar, and this year it's been a bit more stressful in the UK because of the postal service strikes, so who knows if some of the stuff will arrive or whether they will have to be birthday or Easter presents, or if they're really late, I guess, Christmas 2023. I was just editing the events page at Labiotech.eu earlier and taking out some of the ones that have already finished, adding a few that have been announced for 2023 and just doing a general tidy up of the page. There's nothing worse than going to a page that promotes events from 2021. I even visited one recently that had events listed for 2017. I guess that was when the word COVID wasn't even in our vocabulary. Anyway, if you're associated with an event or you've announced 2023 dates and venues, please let us know so that we can add it to the calendar, and also so that I can plan my schedule. Also, if you're interested in having us be there, we're happy to hear from you. Obviously, we can't be at every event, and I promise not to be biased in favour of events in warm climates. Well, I should get to this week's podcast, and we have 3 interviews. We have conversations with Antoine Espinet, CEO of MicrofluidX, Will West, CEO of CellCentric, and Sara Holland, patent attorney at Potter Clarkson. So now it's time for the news you may have missed over at Labiotech.eu. New biomaterial can regenerate bones and prevent infections. Step Pharma announced promising preclinical cancer data, and an antibody discovery could lead to new therapies to tackle Strep A. Not to be outdone by Strep A, we had a story on Strep B as well, as Minervax raised €72 million to develop a Group B Streptococcus vaccine. We had an article on tracking the spread of antimicrobial resistance. Enterome completed patient enrollment for a recurrent glioblastoma study, and OSE Immunotherapeutics announced its latest acute lymphoblastic leukemia data. Japanese research offers hope for Fukuyama muscular dystrophy patients. Poolbeg's trial on influenza was successfully completed, and some research on blood clotting could give hope for treatments of sepsis. Therabest and Glycotope are going to assess breast cancer combination treatment. We have a video interview with Medicon Village CEO Petter Hartman, and Medicon Village is definitely a really fascinating biotech hub in southern Sweden. We had our special newsletter on startups with 10 new articles, not just featuring startups themselves, but also some articles with advice. A Spanish company has been launched to produce melanin from enzymes, a planning application has been submitted for what will be Europe's largest life sciences building, and a spoiler, it's in London, and Precision Life are to work on R&D with Ono Pharmaceutical. NMD Pharma has been cleared for a spinal muscular atrophy trial. Storm Therapeutics closed $30 million Series B financing, and Solve Therapeutics was launched to tackle cancer. A Charcot-Marie-Tooth study is receiving a $250,000 grant. Cell reprogramming can reverse fibrosis after a heart attack, and we had an article on why the EU must refine its ATMP regulation. And you can read all of these and lots more at Labiotech.eu. So now it's time for the interviews. This week was the 64th American Society of Hematology, or ASH, annual meeting in New Orleans, Louisiana. The first time I was there was right after a hurricane, but a beautiful place to visit. There have been lots of press releases coming in from the event, and so we thought we'd do an interview with a company that gave a presentation there. So we talked to Will West, CEO of CellCentric, who spoke to us from New Orleans.
We are a biotech company based in the UK, although we've got clinical trials in North America, mainland Europe, as well as in the UK. We originally span out of the University of Cambridge, the Gurdon Institute, with Professor Azim Sorani. We look at epigenetic related mechanisms, and today we have a small molecule drug called inobrodib that we're progressing from phase 1 to phase 2.
I'm glad you said that word before I had to say it. So I wonder if you could tell me a bit more about inobrodib and what it is and what it treats.
Thank you. Yeah. Interestingly, we've put a link on our website of how to pronounce inobrodib because I too struggled when we first came up with the name. So it's a small molecule inhibitor of twin proteins called P300 and CBP. They are histone acetyltransferases, but actually more importantly, they are activators of certain gene expression pathways, notably MYC, IRF4 and the androgen receptor. And so when you inhibit P300 and CBP, they're two twin proteins, you downgrade the expression of key cancer drivers. So we target the expression of MYC, which is a key oncogene, but as well as IRF4 and the androgen receptor. So in late stage multiple myeloma, disease is very driven by IRF4 and MYC. In prostate cancer, that's more sort of MYC and the androgen receptor driven. So we're using the drug in multiple different settings. Where we've had most success to date is in the hematological malignancies. And that's the reason I'm in New Orleans, because it's the annual meeting for the American Society for Hematology. So yes, we were announcing our clinical data on relapsed refractory multiple myeloma for the first time this weekend.
Is it something that's best used in conjunction with other treatments or as a monotherapy?
I think that's been an exciting thing. So in late stage disease, patients don't respond to other drugs and standard of care. So the number of options left is pretty minimal. With our drug, we've seen clear efficacy on its own as a monotherapy, which is fantastic. We have seen additional benefit when we've combined with two existing agents, so pomalidomide and dexamethasone. So we are likely to progress in clinical trials both as a monotherapy and in combination with PomDex, but potentially other agents as well.
Could you tell me about some of the latest results that you were presenting in New Orleans?
Yes. So when you look at multiple myeloma, you're looking at a number of different indications of whether or not you're having a biological effect. And the key one is measuring serum free light chains and then M protein. In 7 patients that we tested at the recommended phase 2 dose and schedule, 6 out of 7 saw a reduction in serum free light chains. 3 have seen clear responses, one of which what is termed a complete response that then became a long-term, very good partial response. 3 patients have stayed on drug for over 8 months. These are patients that actually just have a few months left, typically. So as monotherapy, we're seeing very clear effects. In combination with PomDex, we've dosed 3 patients, and all 3 have responded within the first month of treatment. Normally that takes 2 or 3 months to kick in, but we've seen a very fast response in the combination.
So is it something that obviously everybody's working towards cures? Is it something that's stopping the issue, or is it just prolonging life? How is it meant to work in that respect?
Good question. I wouldn't position this as a cure medicine, but certainly one that, yes, reduces tumor sizes. We've proven that. And yes, elongates life.
All right, that's good. You're calling from Louisiana because of the ASH event. How important is that in the calendar?
So for people working in hematological malignancies, it is the showcase meeting of the year. There's 30,000 people here. So for us to announce heme data or clinical data for the first time, doing that at ASH was important. We were lucky that our clinical trials were beginning to read out towards the end of the year, and thus, it timed nicely with ASH. But yeah, here we have most of the international hematologists that we would want to share the information with. There's a lot of pharma companies and other associated companies. So yes, it's a great platform.
What was the reaction like to the data that you were presenting?
It's been extremely positive. A couple of reasons, really. I mean, one is the efficacy, but that was kind of in the press release and kind of people were starting to know that. The two aspects I think surprised us. One is that this is an oral agent and people are looking for oral solutions that can be widely adopted in the community. So it's all very well having sort of these super complicated new treatments, but if the vast majority of people can't access them, then that's not really a good solution. And the FDA, for instance, is really focusing on how do we see drugs for the real world developed faster. The second point, which has been really affirming, is the safety profile. We shared in detail the safety that we've seen on the patients that we've seen, both as in dose escalation and then also recommended phase 2 dose and schedule. And actually, for a heme drug, this is a remarkably well-tolerated agent.
And so what are the next steps?
We're pushing forward in relapse refractory multiple myeloma as monotherapy and in combination. We may add another combination. We've seen encouraging results in AML, but that will probably be positioned as a combination with azacitidine and venetoclax, so Venasa, as that's called. The peripheral T-cell lymphoma results are really encouraging, and that may progress further as a monotherapy without needing to go to a combination.
Clearly, what you're working on now is impressive enough, but is there anything else in the portfolio?
We're now wholly focused on inobrodib and P300 CBP inhibition. Our background, as I said right at the start, was looking at epigenetic processes overall. We probably explored over 50 different mechanisms, targets, pathways, actively worked on 7, licensed one to Takeda Pharmaceuticals. But yes, I mean, the one that we've progressed and seen that alignment between really interesting biology, strong chemistry, and then clinical benefit is the thing that we're working on. We are looking at multiple settings, but actually it's just one drug that we're developing.
Next, it's to MicrofluidX, a UK-based provider of next-generation bioreactors for cell research and manufacturing that recently raised £3.3 million. To tell us what the company does and what the funding means is MicrofluidX CEO Antoine Espinet. So I wonder if you could first give me some of the background on MicrofluidX.
Absolutely. So we are a cell culture hardware company. We're developing bioreactors that are connected and have a number of sensors on them as well in order to do two things. First is to really accelerate cell culture research and bring cell culture research in this kind of enterprise 4.0 world where things are more connected, where there's more data flowing and where the data is exploited. And the second mission is really around cell therapy manufacturing. And the idea here is to lower the cost of treatment and make it available for a lot more people around the world.
And can you explain what Cyto engine is?
Yeah, so the Cyto engine is at the core element of MicrofluidX. It's a blend of 3 different components. The first component is bioprocessing. The second component is sensors. And the third component is data. So let me go a little bit into each of these components. So around bioprocessing, as you may know, if you put garbage into your sensor, you get the garbage data out of your sensors. And so with bioprocessing, it's very important to have a precise bioprocessing platform where we have a high level of control over the cells and over the environment around the cells, and also where the measurements that we take in this bioreactor are representative of what the cells are actually seeing. And so that's why we use MicrofluidX, which typically has quite a low volume to cell ratio in order to get that level of process control. The other aspect of bioprocessing is around the scalability. And so, you would argue that a multi-well plate, for example, is a relatively precise bioprocessing platform in the sense that the volumes are quite small and you can change them quite rapidly. But the issue is that it doesn't scale. So if you have a great treatment or a great process in a well plate, it's not going to scale easily to a manufacturing where you can treat a patient. And so you're going to have to go through process development in larger cell culture vessels, and the cells are going to behave very differently at that time, and you're not going to have a precise bioprocessing platform anymore. And so with MicrofluidX, we found a way to scale which doesn't change the environment for the cells, and so that means that you can translate your cell research from a few milliliters of media or cell suspension into several tens of liters, which is more than enough to treat a patient. The second component is around online sensing, and so here, we're working with partners. We're constantly scanning the market for the best online sensors. The industry is quite young in that respect. There are not many sensors that you can actually get, but there are a lot of innovative companies that are coming up with technology that's very interesting to measure what's going on with the cells. So, beyond the typical pH and dissolved oxygen sensing that a lot of bioreactors have, people are clearly really interested in knowing, can you measure viability of the cells? Can you measure the number of the cells without having to take samples? And so that's why we're working with those innovative companies to integrate those onto our bioreactors. And then the third aspect is around data. So historically, a lot of the data has been, paper-based, has been taken on different instruments and then kind of looked at by the user, maybe brought onto an Excel and then kind of looked at by the user. But we think there are a lot of insights that are kind of hidden, and where computers and machine learning and even simple analytical tools, statistical approaches can help uncover this data. But in order for that to happen, all the data needs to be in one place, needs to be formatted in a certain way so that it's easily exploitable. And so, working towards that, and then again, working with third parties that have these powerful analytical platforms to plug into their platform and allow the user to then understand their process better.
What's the current issue with being able to provide therapies affordably, and how are you addressing that? Because I noticed in the press release that came out, it mentions the fact that there are two large pain points, and that's commercialization and clinical translation.
Yeah, I think with the affordable aspect, there are very obvious factors, and there are some less obvious factors. So on the obvious factors is, around removing manual interventions, trying to lowering the reagent cost, trying to lower the cost of the facilities, which is broken down around the grade of the facility. The cleaner, the higher grade facility, the higher per square meter it is to build and maintain, and then the footprint as well, the square meterage as well. So these are kind of the obvious cost, factors that you would think about when you think about, for example, cost of manufacturing, right? Cost of goods of a therapy. And so, with automation, obviously we are an automated platform, and I think a lot of other bioreactor companies are looking at automation. And so this is the biggest lever in reducing the COGS aspect for sure. It has some challenges, but it's relatively straightforward. You put pumps, you put valves, etc., and you transfer the liquid. And so that's what we do as well. The second aspect with reagents, I think here we have a clear advantage being in microfluidics, because we make a better use of the reagents, essentially by bringing the cells and whatever molecules are in the reagents closer to each other. We allow a better transfer and a little better contact between the cells and whatever is in the reagent. This is particularly true with, for example, viral vector, which is one of the most expensive reagents used in, for example, in T-cell processes. And the viral vector is used to modify the genetic makeup of the cells. And so by bringing the viral vector closer to the cells, we essentially increase the probability of a viral vector particle encountering a cell, penetrating the cells. And here we've shown that we can actually lower the cost tenfold of that particular reagent, which is straight up 10K taken off the treatment. The facility one, is not a major lever, but I think once you have addressed the other ones, that proportionally becomes bigger, right? And so here the idea is that first you need to close the system so you can work in a grade D facility instead of a grade B. So that's really important, because there's a 10 fold or cost increase between a grade D and a grade B. And so that's, cost of building, maintenance, all the filters as well that you need to replace quite often. And then the square meterage. And so here we have an approach which is around stacking the bioreactors on top of each other. And so we think we actually can save 1,000-fold on the footprint of our treatments by stacking those bioreactors on top of each other. And because of form factor of a bioreactor is quite thin, larger surface area, but quite thin, it's very prone to being stacked on top of each other, a bit like drawers. These are kind of the obvious costs you would want to address, but there are some less obvious ones that I think are really important as well. One is around the cost of batch failure. So with cell therapies, we see manufacturing runs, right? Even treatments that are commercially available with sometimes double-digit batch failure rates. And so as an industrial product, this is not an acceptable amount. I think you would want to be in the percent or so or lower than that for standard industrial product. And so obviously each time you have a batch failure rate, you're wasting. The patient usually is in critical condition. You're lowering the chances of that patient surviving. And also from a COGS perspective, you obviously wasted a lot of resources making that treatment to throw it away at the end. And the question is, why do we have such a high batch failure rate? And that's why I say it's a little bit less obvious, because it's not necessarily about, more people or about more automation, but it's really about making sure that you have a process that is really well controlled from the beginning. And if you are in a bioreactor where, there is no precise control over the cell environment, or whatever you're measuring inside the bioreactor is not representative of what's happening with the cells, then it's really hard to control that process. And to me, in my opinion, that's why we see some of these high batch failure rates, is that just the process, the fundamental understanding of the process is not there, because the work has not been able to be made up front about understanding that process, because the bioreactor was not right. That's one. And the way we address that is, again, with microfluidics and making sure that we have something that you can do at a small scale, really understand your process, really gather a lot of data about what's going on with your cells, and then scale it up exactly in the same way. The second one, which is also related to that, is the cost of a treatment is not just going to be the COGS or the cost of manufacturing. There's going to be, for pharma companies to make a return, they also need to amortize the R&D, right? They need to amortize all the cost that has gone up front. And those costs are really expensive for cell therapies. I mean, both because the reagents are very expensive, the cells are very expensive, you need a lot of people to do the research, and it takes a really long time as well. The way we're addressing this is by providing automation really early on at the research stage, where you can do your cell culture, automatically, gather data automatically, really have this very high throughput cell culture research, where you're really getting a lot of data, really doing a lot of experiments in a short period of time, and then really scaling that immediately to manufacturing without having to go through that process development. So this is how we think we can also really reduce the R&D cost, which ties to your second question around, commercialization and clinical translation. Things are really slow at the moment, because you have to essentially go through that process of cell, research, which is the focus is always just on the cells, right? These research labs, just scientists, really focusing on how are my cells doing. They're not thinking at all about manufacturing at that point. Once they have figured out, okay, I have some kind of a process that I can do in a multi-well plate or maybe a flask, and I want to take that into, spin out the biotech and go into the clinic, there's a really long phase, can last 2 to 3 years of process development, where you're taking that process that goes from a flask into a bioreactor typically, right? And then as you scale up that bioreactor, you're going to have a lot of problems, because the cells are not going to react the same way to the environment. You're not going to be able to control your environment as precisely as you increase the volume, because typically people don't measure a lot of things during the process. You're only going to be measuring endpoints, right? So you're going to do your runs, you're going to look at how my cells do, And if they are good, then you say, okay, that's great, that's my process. If they're not good, you don't understand what has gone on. You don't understand, is it at some point did they lack a certain cytokine, or at some point did a certain metabolite go up too much and that affected the viability? You don't know because you're not measuring. So what we're doing for that is really offering to scientists a microfluidic bioreactor that's manually operated. So it's very cheap. You can do it exactly the same way as you would do well plates, but you have all the advantage of microfluidics, where you can, keep the volumes quite low, not use a lot of reagents, and have a lot, if cells in largely like that environment. Then we offer automation of those same bioreactors so that you can quickly understand your process, and then we scale up to a dose. The way we do that is we take those research bioreactors that we have offered scientists at the beginning, and we print those exact same bioreactors many times over a larger array, which forms a larger bioreactor. But it's a bit like electronics, because you're printing many transistors next to each other. Every single transistor, it's happening exactly the same thing in every single transistor. And so that's how we scale without changing the cell environment, is by taking those exact same bioreactors that are used for research, but printing a lot of those bioreactors in parallel. So then you can reproduce that process many times and get the number of cells that you require.
You just got £3.3 million in funding. What will you be using that towards?
The funding that we have is really to get our prototypes in the hands of end users. So the money is to build those prototypes. We're very quick at iterating prototypes, and as soon as we have something, get it in the hands of users. So we had a press release about a partnership with STEM Matters last month, and we poised to announce quite a few more coming soon. And so the idea is that we have a fully automated closed system prototype of our manufacturing bioreactor, which can produce billions of cells, has integrated analytics. He has PhDs of oxygen, but also a very good imaging system that is able to look at how many cells are in your bioreactor. And so the idea here is that we want people to try it and give us their feedback. It's not going to be perfect, but we think it's going to be really advantageous for the cells. And then in parallel, we also have our research prototype, which is the research bioreactor that I mentioned earlier, which is operated semi-automatically. And this is the same story. It allows us to trial things quickly with end users, see if their process could fit in our platform without spending a lot of time and a lot of money. And then if they're happy with that, we can move to the larger manufacturing system. So, we have explored T cells, we have explored mesenchymal stem cells, and we're starting to work with iPSCs. And so, going through that research platform first, knowing that it works, then we can scale to the manufacturing platform. And so, yeah, that's what we're going to be doing in the next months with end users.
So what does the timeline look from here?
So next year, we'll be focusing on those end user trials, and we'll come out with our semi-automated, semi-manual, depending how you look at it, research platform. In the meantime, that will be available commercially next year. And then we'll keep working, keep iterating the prototype of our fully closed, fully automated manufacturing platform. It requires a little bit more work. It will come most likely as a research use only in 2024, and planning to go to GMP towards the end of 2024 with that manufacturing platform.
And now it's to something that most companies need, but might not necessarily know about, or if they do, might not know how to deal with, and that's patents. But the conversation isn't just about IP and patent law, because Sara Holland, who's a patent attorney for the company Potter Clarkson, is also one of the founders of the Women in Synthetic Biology Network. So we chatted about that too. First, if you could start by giving me a bit of an overview of what you do and how being an attorney in relation to biotech companies works.
Yeah, okay. So if I just give you a little bit of my background, then you'll know why I guess there's certain things I like about the job that I do. So I was a postdoc for 8 years. So I was at university until I was 33, basically. working in molecular biology research, all at Nottingham University. And then I realised I was not going to get my own lab, so I needed to find something else to do. So found this as a career. And I don't know, it totally suits me. I love it. I feel much more like a scientist doing this job than I ever did in the lab, which feels weird, but I think when you're doing scientific research, I think quite a lot of the clever and the interesting part is in coming up with the experiments and the ideas. And then you just have to spend days and weeks actually carrying it out and getting the lab work, getting the results, which to me, after 8 years, I found that part to be quite boring. I think I work on lots of different technology. I work all the clients I work with, it's all biotech, but it can be, biotech's quite a big, broad thing. So it could be engineered microbes to make oil and then antibody therapeutics. So it's all biotech, but very different. And that variety I really like. As a patent attorney, a key part of my job is to secure IP rights for companies, mostly patent rights. And most of the types of companies I work with are very early scientist founded companies. So they're very early startups. Some spin out some universities. Some universities I work with directly with their tech transfer office. A key part of my role is to secure these patent rights for these companies so that they can build their business, because without the IP, you don't really have a viable business, especially in biotech, because investors want their money. They want to get the return on their investment. And I think after spending so long in academia, it's just not something you think about as a postdoc or a PhD student. You're there, you've got your project, you're funded to do a specific bit of research. You somehow, and I was very guilty of this, think that there's some magic commercialization fairy, that you're going to publish your research in Nature, hopefully, for those that make it into Nature, publish it, somebody's going to read it and think, oh my God, this is amazing, I'm going to use it to cure cancer or save the world. But I very quickly learned as soon as I left the university really and started this job, that's not how the world works. And if you haven't thought about protecting your IP, you're just not going to get the money to get your business off the ground. So as part of obviously my day job is to support these clients, but part of what I like to do as well is some IP education, because I know there's a lot of people like I was in the university who've got all these really good ideas, but they're not thinking along the right lines and they're just publishing it, and then society misses out basically because all these good ideas just don't get translated. So that's one thing I'm quite keen on, trying to get work on this kind of translation piece of getting research from universities or from these very early stage companies and helping them get their technology out into the world so we can all use it. Because I'm not knocking some industries, but we all like a nice new TV, but that's great. But the kind of stuff people are doing in biotech, it has potential to have a real impact on climate change, health, food security. So I guess that's where I see my fulfillment in my job, because I'm playing a tiny, tiny little part in helping some of these companies succeed, really.
Just listening to you there talking about companies, I guess when, like you said, when somebody starts a company, they don't really know anything about anything other than the research. How do you make that connection in terms of how do they find you? Or because obviously when they start a company, they don't think, oh, got to find a patent attorney.
I suppose historically, getting something patented is a bit of a tick box exercise. People have probably historically known, oh, I need to look at IP at some point, but they don't really value it and don't really understand why it's so important. I personally go to a lot of fairly academic conferences because of my interest in the academic education side of it. My own personal focus is in the synthetic biology space. So I go to a lot of synthetic biology conferences, meetups, talk to people about, I don't know, don't really go in, diving in and talking about IP. It's more I'm interested in what they do. And then at some point they realize they do need a patent attorney. I think as well, increasingly, there's more, a bit like Entrepreneur First and Carbon 13, where there's a group of people looking to found a company. They come together and meet co-founders. They go through a bit of an accelerator program. So they are kind of taught a little bit about, you need to think about IP, thinking about raising finance. So I think they are increasingly becoming aware of the things that they do need. But yeah, I think founding a company, you are kind of very much on your own and you're looking for somebody to support. And I think to support you, sorry. And I think that's the kind of relationship I like to have with a client. Because I understand me asking a client who doesn't really know much about IP to make quite a big decision about their IP is probably not very helpful for them. So I think as a firm, we kind of like to guide people through and help them make a decision rather than just, oh, it's your decision. We're kind of waiving all liability for this. It's on you. That's not how it's supposed to work. And I think having that kind of close relationship helps us to be a bit more proactive. So I think there are some clients who want the more traditional arm's length. They know what they want. They tell us to do it and we do it. But the kind of clients I'm working with don't know what they want or what they need. And what they need is something that a proper, thought out, well-planned IP strategy that supports the business to take them from where they are now to where they want to go. That's going to help them raise the investments. Like we said, you're not going to make an impact in the world if you don't get the investment to get your company going in the first place. So I also do a lot of posting on LinkedIn. That's where a lot of my contacts come from, on LinkedIn, because I do a lot of, again, the IP education side and just generally trying to raise awareness of what biotech can do, because I know I'm guilty of being in this little bubble of everyone I speak to knows how great biotech is. I know there's a lot of people out there who don't know anything about biotech. They think synthetic biology and GMOs are evil and something that we all must run away from. And it's kind of, I don't know. I'm just one person with this like little tiny platform on LinkedIn, but I want to at least try to spread the word.
Right. How important is it for companies to tackle IP early?
Oh, very. Ideally, speak to somebody as soon as you think about setting up a company, because you don't necessarily need to have a patent application on file. Filing a patent application might be the wrong thing to do. You might want to think about keeping something as a trade secret, but you can't go back and rectify a mistake. So if you start a company, you know literally nothing about IP, you might have a whole bunch of IP ownership issues. You might be using somebody else's confidential information. You might publish something, especially if you're coming from an academic background. Your whole life has been to publish. But once you publish something, you can't protect it with a patent generally. So speak to somebody. Most companies will give you a good chat, at least for free anyway, and put you on the right path. But yeah, get some advice early on, because you can't go back and sort things out. And the stuff you do, the kind of fundamental IP that you're developing early on could be crucial for the whole of your business. So.
Right. And are there any sort of international implications? I mean, do you have to deal with international issues of borders and different countries?
Yeah, so patents are territorial. So if you wanted to get patent protection in the US, we need to file a US patent application. Same for China and Japan. And most of the laws and rules around patentability are similar, but there are some key differences that you need to be aware of. For example, one thing that comes up in the synthetic biology space a lot is, and people still aren't aware of it, in Europe, you can patent protect a naturally occurring microbe, an engineered microbe. You can patent protect a DNA sequence in Europe and in most other places. But in the US, that is very hard to do because of a Supreme Court decision a few years ago. In the US, you can't protect naturally occurring products. So you couldn't protect a natural yeast strain, whereas in Europe you can. So all this kind of needs to feed into your IP strategy. And another key difference is that in the US, there is a grace period for prior art disclosures by an inventor. So like I just said, if an academic publishes their research, generally you can't get a patent to it then because it's not new. If you publish it before you file a patent application. But in the US, there is this 1 year grace period. So if anybody's listening who has published something in the last year, there is still a chance you could get something in the US. So it's not totally over, but you are limited in what markets you can get protection in them.
Right. So not only do you need to know what the laws are in all of the different countries, you also kind of need to know what the strategy of the company that you're dealing with is, if US is a priority or what their priorities are.
Yeah. And for some companies, the US is enough. That's all they want. And it is obviously very expensive. When you get to the stage in the patent system of needing to decide which countries you want to go into, it can get very expensive. So most people don't go for more than 4 or 5 countries because it adds up. So some companies, the US is enough. Mostly it's US, Europe, maybe China, Japan, for UK originating clients anyway. So yeah, you need to have in mind to where people are going to want protection.
You mentioned that it's expensive. Does that mean that companies should get financing first before they tackle that? Or is it one of those kind of what comes first, chicken or the egg?
Yeah, it's hard to start to know, especially with the kind of clients I work with that are they've got no or very little money, but they need the IP to raise the finance. So I think one thing that companies can do early on, and when we speak to investors, it can be more attractive even than having an application on file, is having a well thought out written IP strategy. So I think investors like to see that the founders have some idea of what they're talking about. They've got a clear plan. They know, even if they've not got anything on file at the minute, they know what they're going to file on. They know what they're going to decide to keep as a trade secret, and they know why they're going to do that. So they've got reasons for their planning. And that's the kind of things that we can help with early on. And I've done that with quite a few clients now. We work with them for a fixed price, get them an IP strategy, and then they can put that in their data room to show investors, we haven't got anything on file yet, but this is what we're going to do. Or we have got something on file, and this is our like longer term 5-year plan of what we're going to do. So that does help, I think, for them to get the kind of pre-seed and seed funding. In terms of by the time you get to the point of having to decide into which countries you want to go into, that's 3 years from the first filing. So you've got this 3-year kind of runway to get investors on board, raise the cash to cover those costs. I think that's kind of a typical approach that companies take. Is what universities tend to do as well. So university originating IP, universities will cover the cost of the kind of first year of a patent life. And then before this point where things get really expensive, they would hope to have a licensee on board. And if they haven't, then that's when things get dropped. So there are clear, definite points in the kind of strategy where things will cost a lot of money. And that's when you want to make sure you've raised your funding.
Are there any other challenges that startups and spin-outs have that you can maybe give some advice on?
I think one thing that comes up a lot is the IP ownership issues that people aren't aware of, or they go into set up a company without having thought, well, where's the IP that I'm building this on originated from? Because a lot of founders, especially in biotech and synthetic biology, are building on something they've either done at university or are still in at university and bringing out into the world. IP ownership in the UK anyway tends to reside with the employer. So I think there's quite a common sad misunderstanding on academics' parts that they think they own the IP, but really it's the university that owns the IP. So there's a whole lot of things needed to sort out there. If you developed the project or your business with somebody else, then they might have a claim to the IP on that. So pretty much all the very early stage companies that I speak to, we can do the IP strategy thing, but the thing they need most immediate help with is sorting out IP ownership. The problem is if you don't sort it out now, while your company is valued at very little, in a few years' time, when your hopefully company is like on the way to being a unicorn, that's when people are going to want to come after you, when there's something worth it. So the advice is sort it out now, even though you might not want to get the tech transfer office involved, you might not want to reach out to your past collaborators. It's very important that you do it now, because investors will ask you and they will want to know who owns this IP. And if you can't give them a clear answer, then, there's been cases where people just walk away because it's too risky.
What do you think about the scene right now in the UK for startups and the spin-out sector? Is it healthy?
From what I've seen, yes. I know we're in like a hard economic times, but I think, and from when I've spoken to some VCs, the early stage funding, it's still there. So there's still a lot of activity in the pre-seed, seed funding, the founding of companies every time. Because I go out quite a lot to a lot of events, I feel like I've seen all the companies, but there's always more and more coming, all biotech companies all over the place. So I think so. I do wonder what's going to happen more longer term, because a lot of the companies are doing similar things like strain engineering, enzyme engineering. A lot of people in the cultured meat space, are they going to end up consolidating, or I don't know. Don't know what's going to happen more longer term, but at the minute it seems buzzing.
It's a good thing. All right.
Yeah.
And you're involved with the Women in Synthetic Biology Network. Could you tell me something about that?
Yeah, so this started from a recognition between me at Potter Clarkson, Liz Fletcher at the Industrial Biotech Innovation Centre in Scotland, and Fiona Michelle from SynBioBeta. We kind of all collectively at the same time realised, well, for me, I don't work with very few female inventors and female founders. Fiona organised a synthetic biology meetup in London, and I think she said there was like 3 women compared to 50 men. So we thought we'd just put together a network for women working in synthetic biology, or kind of loosely associated synthetic biology. We've had a London event, an Edinburgh event, and a Manchester in-person event. And then every month we've been trying to hold an online session where I think we want to try and create a safe space where women can come, ask whatever question they want to ask, find out from people who are happy to give their time up and give something back. Because like we said, especially if you're founding a company, you've got to do everything. You've got to be HR, finance, IP. And I just can't imagine being in that position. I often wonder, looking back at my academic career, thinking, why did I never think about setting up a company? And I just don't think it was a thing that people really did 10 years ago. But even now, I don't think I would have the guts to do it. So all these female founders, it's are awesome. And we want to do what we can to help them. So yeah, so hopefully we're having another in-person event sometime early next year. And yeah, it's like one of those things you start and then you don't know if there's a need for it or people care, but people do seem to enjoy it. So, and I think you do have different conversations as well.
Right. So it's more of a support network. It's not just people that are in synthetic biology. It's sort of ancillary businesses as well that can help them out.
Yeah. So we've had people, grant funders speaking, VCs speaking, some founders talking about their experience of founding companies, just, what is it actually like as a woman founding a company in biotech and synthetic biology. So
you may be able to have to start an international section soon.
Yeah, well, the online things we've had people from India, America, all over the place just turn up to the online ones, and that's quite nice. So we normally have a half-hour talk and then break out into little rooms so we can all just chat to each other.
And that's it for another week. There will be a podcast next week, hopefully, but not in the week between Christmas and New Year, so I will be able to wish you happy holidays then. This week, I guess it's just happy shopping, and as well as wishing you well, I also hope that you get a parking spot close to the stores. So I hope you enjoyed this week's podcast and that wherever in the world you may be, you have a great week ahead, and join us next time for another Beyond Biotech.